Feindel virtual Brain and Mind lecture. I'm really thrilled today to have Shella Keilholz here as our presenter. Shella received her PhD from the University of Virginia in Charlottesville in engineering physics. And then she progressed to have a postdoctoral fellowship at the National Institutes of Health in the National Institute of Neurological Disorders and Stroke with a focus on neuroimaging. She started as an assistant professor in the Department of Biomedical Engineering at Emory University, which I understand is also joined with Georgia Tech University in Atlanta. She was promoted to the rank of associate professor in biomedical engineering in 2014 and is now program director of the 9.4T animal scanner at Emory. And she's been in that position since 2015. Shella has numerous publications in the field of neuroimaging and MRI. She's a handling editor of Neuroimage and on the editorial board of Network Neuroscience. She holds multiple project grants from the National Institutes of Health as well as the National Science Foundation. And all of this work is very innovative and quite remarkable with a broad goal really to develop methods for mapping spontaneous activity throughout the brain at an ever higher spatial and temporal scale. And really some of this aim is to understand the dynamics of functional networks that are underlying bold signal. And I'm sure Shella will be sharing so much more about her work today, and I'm very excited to hear it. Before we jump off and get started, I just ask that everyone remain muted throughout the duration of this talk. If there are questions, please feel free to put them in the chat box. If there are issues regarding clarification, we may interrupt the speaker midway through to do so, but we prefer, in fact, the questions be held until the end. And if they be entered into the chat box, I will then read them to Shella at the conclusion of her talk for a more extended discussion. So thank you all again very much for being here. This is wonderful. There's so many of you here today. Shella, please take it away. I look forward to your talk.
Thank you so much for that really awesome introduction and for the invitation to be here. It's always a treat to talk to people about science, but I think it seems especially nice when we've been confined to our houses and out of the lab for a while. So I'm really looking forward to having a good discussion after this. I've tried to keep the presentation short and to the point because I'm hoping to hear a lot of questions and have a lot of kind of feedback and ideas about where this might go. So the title I picked today is Functional Connectivity Dominated by Neuromodulation? And, you know, just as kind of a spoiler, I think it is. We don't have exact proof of that yet, but I think it's time to start thinking about some of the implications. So I'm sure for this group, y'all are familiar with resting state functional MRI. It's become very popular because you don't have any task or stimulus. And so you can do comparable studies in patients who don't cooperate, in people who are asleep or anesthetized, in animal models. And look then at the intrinsic organization of the spontaneous fold fluctuations that we think reflect some sort of aspects of the coordinated neural activity. So the most traditional way to look at these fluctuations then has been functional connectivity analysis. And I'm going to call it average or static functional connectivity because this was traditionally calculated over the course of the entire scan. So you might take a seed region or a pair of ROIs and then calculate the correlation either between that seed region and the rest of the brain or between different pairs of ROIs. And that would be performed for the entire time course of the scan. So usually 5 to 10 minutes. Now people realized, you know, for a long time that the brain's not doing the same thing over those 5 to 10 minutes that you're in the scanner. At least we hope not. And so people started to look at dynamic or time-varying functional connectivity. And the simplest version of this is really to take, instead of an entire scan to do the analysis on, some sort of windowed approach. For example, sliding window correlation just takes a chunk of the scan, maybe 100 time points, and calculates correlation in that window and then moves to another window. You can look at the kind of the same data at the individual event level, where you then take the time course and look for individual times where areas pass a threshold, and you can produce these coactivation patterns out of those.
There's hidden Markov models, there's clustering, there's dimensionality reduction. My lab has played with all of these at some point, and I'm happy to talk about those later if you all are interested. But what I really wanted to focus on today is what I consider the big question in resting state fMRI, which is what gives rise to the coordinated patterns. When I started playing with resting state fMRI, gosh, ages and ages ago now, I guess it's been about 15 years, I was convinced that it was all noise, that it came from vascular effects, from respiratory effects, cardiac artifacts, and definitely these processes contribute. We know that they can all influence the signal that we pick up in different areas of the brain and give this pattern of correlation that may be artifactual. But on the other hand, at this point, we're confident that this is not the only thing that we see in resting state fMRI or in functional connectivity. My lab has done a lot of multimodal studies, and we see very nice correlation between the coordinated fluctuations that we see in the bold signal and the coordination of the underlying neural activity. And there are many other labs who've gotten similar experimental evidence of this. So then, so that's kind of one view. Another sort of similar idea that maybe this was related to artifacts is if you go to time varying functional connectivity. A lot of people thought, well, maybe this turns out to come from properties of the signal itself. Now this is something that we were worried about. So if you're looking at changes in functional connectivity over time, maybe it's because your signals themselves are autocorrelated, have certain frequency patterns, and so you pull out these spurious fluctuations. But just like the idea that perhaps physiological noise is really driving functional connectivity, I think with this dynamic sliding window analysis for time varying functional connectivity, we can say the same thing, that the artifact is there, but it's not the only thing that's there. So we still don't know where these coordinated patterns come from. One of the original ideas was that if you had strong functional connectivity, it represented some sort of direct functional connection. And that holds true for many areas of the brain, but not for all of them by any means. So some areas like the caudate putamen, for example, are very weakly or not connected at all directly from one hemisphere to another. And they show correlation just as strong as areas that are directly connected through the corpus callosum, like somatosensory cortex. So that doesn't really explain everything. Then we thought, I guess kind of the hope was when we started looking at this time varying functional connectivity, taking it to the other extreme. If it's not an artifact, maybe what we're really looking at is this true variability in neural activity over time that's related to cognition, to different brain states, and to other things that are going on. But I think with all of these artifacts that are present, that has to be taken with a grain of salt too. So none of these kind of one-track explanations has held up. And I think what we really need to do is to think of this as at more of a systems level. And so for this talk, I want to talk about something that people don't usually use for functional connectivity analysis, but that my lab has been kind of hung up on for a while. And that is something we call repeated spatiotemporal patterns. We also call them quasi-periodic patterns, or QPPs. So let me explain what these are and why we found them first. They are a repeated spatiotemporal pattern that occurs in the background almost constantly during these resting state scans. So a repeated pattern, it follows the same timing, the same areas are involved, the same pattern of activity moving from one area to the other, going on in the background all the time. Now this is not something we were looking for. We were really acting on this hypothesis that time varying functional connectivity should reflect variations in the brain that are related to something like cognition or information processing. So we took some very simple steps when we were first trying to look at this. And we found that if we just took resting state data and kind of normalized the signal values, so we got rid of any outliers, we could see repeated patterns of propagation from the lateral cortex to the medial cortex in anesthetized rats. And we thought it was an artifact, so we tried looking at dead rats, we tried phase scrambling the data, we did all sorts of stuff, and we couldn't make it go away. So we thought, well, maybe it's just something that you see in anesthetized animals, and maybe that's why there's this repetitive activity. So while we were trying to characterize that, we developed a pattern finding algorithm that makes a template of the type of activity that keeps going on in the background. And just for kicks, we threw in some human data, and out popped these same sort of repeated patterns in data from awake resting humans. And moreover, in the humans, they involve the default mode and the task positive network, which are of a lot of interest for many different reasons. So it makes us think that these are important. So first, I'm just going to show you what one of these looks like. This is in the Human Connectome project data. So you can see here, if you look at the color bar, the warm colors are things that are above the mean signal. Cool colors are below the mean. And I'm going to play the movie. What I want you to notice is that this really involves the whole brain. We see it all over the cortex. We see it in the cerebellum, it's in the brainstem and the thalamus. So this is really a systems phenomena. It's not something that's localized to one area. So now that you know kind of what it looks like, let me describe this pattern finding algorithm just briefly. So what we do is we take a functional scan over time, and we pull out a random chunk of data. We know in humans that the chunk ought to be about 20 seconds long. And so we just select a chunk that fits that criteria. And then we do correlation between that chunk and all possible chunks of the resting state scan. So over here on the left side of the screen, what you see is this correlation time course. And there are peaks where there are chunks in time where the spatial pattern and the temporal timing of the activity looks like the chunk we randomly chose. So we find those peaks, we pull up the chunks that correspond to those peaks, we get the images over time, and we average them all together to make a new template. And then we repeat that process until that averaged image, which we call the template, doesn't really change much. And so what we get out of this algorithm at the end is this 4D template, which is volume over time, our 20 seconds worth. And then we have a sliding window correlation vector that tells us how strong that pattern is at any given time during our scan. Okay, so I'm just going to play this again now that you know how we found it. And so one thing you can see is that the default mode network and the task positive network are anti-correlated. I'm going to play that again. And if you look at things like the cerebellum, areas of the cerebellum that are generally active with the default mode network are active with the default mode network in this pattern as well. And the same goes true for other areas if you look at known connections and known functional connectivity mappings. So I'm going to zoom in here so you can see the activity in the subcortical structures a little bit more. It's very interesting. So there again, you can see, in the cerebellum, but you can also see that, parts of the thalamus tend to activate before other parts, and there's sort of this propagation within the subcortical regions as well as within the cortex. Okay, so we wanted to kind of compare this way of looking at resting state fMRI to some of the other ways that are out there. And one of them that immediately caught our interest was the paper by Daniel Margulies from 2016, where he described functional connectivity gradients. And if you look at the gradient 1, which is the top one at the left, what you see is that it looks very much like one of the phases of our QPP. Task positive mode and default mode are very anti-correlated. And in fact, if you watch the QPP play out, it basically sweeps from one end of the gradient to the other and kind of makes a cycle. So I thought this was really cool. Daniel had done this great work and showed that, primary sensory and motor areas were at opposite ends of this gradient from association areas. They have the greatest topographic distances between them, and they're at opposite ends of the developmental axis. So there's really something fundamental kind of about this organization. So it's neat that the QPP that we're seeing is picking that up and actually going over that in a dynamic way. Now you'll also notice that there's multiple gradients over there, not just the first primary functional connectivity gradient. And one of the things we've been wondering for a while is whether there are multiple QPPs. So it turns out there are, and what we did is we took our resting state data, we regressed out QPP1, and then we repeated the pattern finding algorithm and got QPP2, etc. So what I'm showing here from top to bottom and left to right are the first 6 QPPs. Now you can see that they're getting kind of noisy by the time you get down to 4, 5, and 6, and I'm only going to talk about the first 3 for the most part today. I think we have to figure out when it makes sense to stop, trying to pull these into patterns. But again, they show the same sort of alternation of higher activity, lower activity, and then some propagation along the cortex. And so here are the first 3 of those lined up, QPPs on the top and functional connectivity gradients on the bottom. And you can see that it does seem to hold that if you go to additional QPPs, they sweep out, the secondary and tertiary functional connectivity gradients. So we also wanted to see just how much these QPPs contribute to functional connectivity. And so I mentioned we regress these out. So basically the way we do that is we take the template, we convolve it with this time course of correlation strength, and then we regress that from the original signal. So this is data from the Human Connectome project. And if you look at the top right matrix is the functional connectivity between different networks, sorted and labeled at the bottom there, things like ventral, somatomotor, dorsal attention, etc. And top right is before we've done any regression of QPPs. Bottom left is after we regress the first QPP. And so what you notice is that the activity within blocks, let's see, I don't know if you can see my mouse, but within a block, the activity tends to be decreased after you regress out to QPP. And this is especially true for areas of anti-correlation. So you can see here that this anti-correlation is much decreased after you regress out the QPP. We can take out 2 and 3. And by the time we take out the third QPP, you can see there's actually very little functional connectivity left. The noticeable exception being these sort of diagonal lines, and those correspond to connectivity between left and right areas in different hemispheres. So like left motor cortex, right motor cortex, left visual cortex, right visual cortex, things like that. But those are still very strong, and I think that's really interesting. So at this point, we found that QPPs are a parsimonious way to describe our resting state fMRI. Taking these 3 patterns account for much of what we see in the signal, but we still don't know why they're there. So my lab is really into doing multimodal studies, and one of the things we've been working on for a while is combining fMRI with microelectrode recording, which is tricky for many reasons. I like to show these images on the left. The first one, the one at the top, you can clearly see an artifact from the electrode. That was one of our early ones. The one at the bottom has 2 electrodes implanted, and it's much harder to figure out where they are. So we're really proud of the image quality there. And we also get some noise in our LFP recordings that we have to handle, but I'm not going to go into that right now. So we did some of these simultaneous imaging and recording experiments, and then we looked for coherence between the MRI signal and the really slow electrical oscillations, so the ones below 1 hertz that are directly comparable in frequency. And we found it, which is not too surprising. It made sense to us that we should be following something that's present in the neural activity itself. I'm showing 2 different anesthetics here. The one on the top, I seem to have lost the labels, but the one on the top is isoflurane. The one on the bottom is dexmedetomidine. And under either condition, we see very nice coherence in the low frequencies. The actual range varies a bit depending on the frequency that we use. Okay, so we can also take the signal from the electrode and do correlation at different time lags to account for the hemodynamic delay with the signal from all of the bold voxels in the image. And that's what we've done here. And what I want you to see is especially visible in the dexmedetomidine data, where you can see that we have peak correlation. Even though we're looking at the correlation between the signal from one electrode, we see correlation on both sides, like you'd expect from functional connectivity. And if you wait a while, it goes from being positive to negative. And then here's hints that it's going to come back and be positive again. So this is kind of evident of that sort of cyclical alternation that we saw in our QPPs. So we looked then at the actual images, and so I'm going to play a little video here that shows the correlation between the signal from the electrode and voxels throughout the brain first, and then after that it's going to be the template that we got just from the bold signal, and they're very similar. So here comes our correlation between electrode and bold voxels with varying time delays. So you see first it's negative, then you see this high correlation moving kind of lateral to medial, and then negative correlation moving lateral to medial, then maybe a hint of it coming again. Now here comes the same rat, data from the same rat, but now made from the bold signal alone. And you can see that the spatial patterns and timing are very similar to what we got from the Infraslow activity. So, you know, we think this is pretty good evidence that we're probably looking at how the Infraslow activity propagates along the cortex. Now, we've tried to do this in humans with direct current EEG MRI. It's hard to get those really low frequencies while people are in the scanner. And of course, the EEG signal doesn't give you the same sort of spatial localization that you get with a microelectrode. But if you look at, let's say, this particular slice here, and we've got time going vertically, this is at a lag of about 4 seconds between EEG and bold. And if you look again at this lag of about 16 seconds, you see that what was highly correlated is now anti-correlated. So we've got hints of that same periodicity, which is not conclusive maybe, but it suggests at least that the same sort of patterns are present in the human data, and that we're still looking at this sort of infraslow activity with our MRI signal. So this is great. QPPs are linked to infraslow activity. What we didn't realize when we first started doing these experiments is that nobody really knows where infraslow electrical activity comes from either. So we've tied one thing that we don't understand to another thing that we don't understand, which is informative, but it's not helping us get to the bottom of the mystery. So how do we get to the bottom of the mystery? Why do we have this coordinated pattern? And you know, this is what I'd really like to hear people's thoughts on. We've got two ideas really. One is that we know that the brain has this structural network. So over here on the right, I put some examples. You can take the brain, divide it up into areas and call them nodes, and then decide on how strongly they're connected based on how many DTI fibers run between them, and kind of build the structural network. And, you know, just having some sort of structural network suggests there might be some sort of resonance, that maybe particular types of activity, you know, propagate at certain speeds or in certain ways across this network. So that's one idea we had. The other idea, after talking to some of our neurophysiologist friends, is that maybe some of these small nuclei with very widespread trajections are actually kind of driving or organizing, or at least occasionally coordinating this pattern. So, you know, maybe we're getting some sort of neuromodulatory input from deep brain nuclei. So I'm going to show you a little bit of evidence that we have for both of these explanations. So, you know, to look at this first possibility that we're looking at a resonance of the network, we took some human connectome project data, we parcellated the brain into 66 different areas, built the structural network based on the fiber tracks between them, and then drove each node with some differential equations that represent different neural mass models. And then once we had this going and parameterized about the same way that people who used it for other studies, we just let it run for a while, and we did QPP analysis on the output. So when we did that, I've got a few different examples here. if we do QPP analysis on empirical data, it looks like this. Now, this is a simplified view. Each line here is an ROI, and then over time, we see it going above baseline or below baseline. So it's not projected on a brain here. This is just kind of to let you compare quickly across different conditions. So we tried two different sets of differential equations for the neural mass model. We tried Kuramoto oscillators, which are basically kind of little oscillating systems that can synchronize, and then we've also looked at just shifts in firing rate. And both of those, this is Kuramoto oscillator, this is firing rate, both of these give us kind of this two-state solution where we get kind of this anti-correlation between things that kind of look default mode-ish and things that look CAS positive-ish. But they don't have that complex structure or the delayed propagations that we see in the empirical data. So we think that these, you know, the QPPs may have some tie to the structural network, but it doesn't explain everything. Part of it may be because these structures only include white matter connections and not direct connections through adjacency of the cortex. But we're not sure yet. We think this is maybe a part of the explanation. So then to get into something I've got a little bit more to talk about on, the neuromodulatory systems, we looked around, and there have been many, many studies where people have looked at pharmacological manipulation of serotonergic, cholinergic, et cetera, signaling, and they see differences in the functional connectivity strength, clustering, modularity, things like that. So we know that these neurotransmitters can influence what we see. We also know that these systems are involved in modulating arousal levels, and something that we knew, but that I haven't shown you, is that QPPs change when animals are anesthetized compared to when they're awake. So that kind of links to arousal. We decided to focus on the locus coeruleus, partly because it's involved in task performance and linked to trial-to-trial variability, which we know is also true of the default mode network, which is a major component of the QPP. We know that the QPPs are tied to infraslow electrical activity, and stimulation of the LC alters infraslow activity in the cortex. We also know that the LC is the site of early alterations in things like Alzheimer's disease. And again, something I haven't showed, but that we knew, is that if we look at mice in Alzheimer's disease model mice, the QPPs give better classification than standard functional connectivity does versus control. So it suggests that, you know, there's something sensitive to some of these early degenerations. Okay, so one of the things that might make sense then is if these deep brain nuclei are really driving the QPPs, we might see some sort of difference in timing across areas. So what I've plotted here is this is the cycle of the QPP. It goes from 0 images to 30 images, which is about 20 seconds. And at each time point, there's a bar that represents how many voxels are peaking in that area at that time. And the black lines are the dip, the lowest point of the left posterior cingulate and the highest point of the left posterior cingulate for reference. So there's a lot here, but what I want you to look at in particular are the thalamus and the deep brain nuclei. And they tend to have a lot of voxels that peak well before the left PCC. So that suggests that these systems that are involved in neuromodulation and arousal may be changing earlier and possibly driving the changes that we see throughout the cortex. So I mentioned that we know the locus coeruleus is involved in task performance. So one easy thing that we wanted to do was look at whether QPPs are altered during task performance. So we first looked at a psychomotor vigilance task, which is what we just happen to have on hand. And that's where people watch this really teeny dot that occasionally changed color from purple to blue over an hour's worth of scanning. So it's really hard to pay attention to this. And when there was a color change, you had to hit a button as fast as you could. So we measured reaction time. So we looked at resting compared to doing this PVT task, and we looked at how strong the QPPs were and how often they occurred. We didn't see any difference. But when we looked at reaction time compared to when, based on when that dot changed color during the QPP, we found that the phase of the QPP had a big effect on the variability of the reaction time. So if you were in the phase where the DMN was active, you had a much more variable reaction time than you did when the task-positive network was active, which again fits with previous studies for people who were using average functional connectivity. So I don't have the graphs here, but we also have recently looked at some HCP data from 2-back-task and 0-back-task, and there we saw that the QPPs are stronger and more frequent than at rest. And we think that's because people are more engaged in these tasks. It's not just trying to maintain attention for a long time, they're actively doing something. So another thing that we thought might tell us something about the sources of these QPPs and these large-scale patterns of activity is whether they are altered in specific disorders. And we chose to look at attention deficit hyperactivity disorder because it's known to have reduced noradrenergic input. Patients have deficits in attentional performance, and we know that the networks involved are related to attention, and they've exhibited altered infraslow electrical activity. So it all ties to the QPP pretty nicely. So when we looked at this, what we found is that the patterns themselves look very similar across the control groups and the ADHD groups. And what we did notice, though, is that if we looked at the time course between the components of the default mode network and the components of the TPN network, in the controls, you saw fairly strong signal changes and big differences between the two networks. And this was reduced in the patients with ADHD. So we did a little bit more analysis. We looked again at the strength of the QPP and how frequently occurred in controls and in ADHD, but we didn't see a difference. But when we did our QPP regression and calculated how much functional connectivity changed after that regression, there we saw something very striking. So I should have put up the original graphs here, but these are organized into task-positive network and default mode network. And so what you see is in the control group, after we regress the QPPs, the correlation between default mode network and default mode network, so within default mode network, the correlation goes down because we're taking out a lot of the common signals, and the correlation within the task-positive network goes down, and the anti-correlation between the two disappears. That's why this is positive. So basically, the correlation within the networks and the correlation between the networks all kind of moves towards zero, which is what we saw before with the QPPs removing the functional connectivity. When we looked at what happened in ADHD group, is we saw much less effect. It's like the QPPs are not contributing as strongly, and that kind of fits with them having a smaller amplitude in the ADHD group. So because the QPPs account for less of the functional connectivity in the ADHD group, we think this is consistent with the idea that maybe they have this lower level of noradrenergic input, if these are really reflecting, input from the LC. So our final test was to use some rats where we can go in and really just kind of do a brute force intervention and kill off most of the locus coeruleus neurons. So if you give this drug called DSP-4, and wait a couple of weeks, the number of neurons that's left in the LC is something like 10 to 20% of what's there is there originally. Histochemistry, and saw that we did get a nice decrease in norepinephrine throughout the brain with not much effect on the dopamine. But what we were really interested in was what happened to the QPPs in these animals. And if you look at these time courses, so this is correlation over time for control animals, and then for DSP-4 animals, you can see that the correlation over time is much weaker in the animals that have been treated with DSP-4. And in fact, if we looked at the QPPs, we just really couldn't see any organized activity. They just kind of looked like noise. There wasn't much there. So we think that, killing off these neurons really did disrupt this organization. Now, I don't mean to imply by this that we think the locus coeruleus is the key area for all of this. Many neurotransmitters affect functional connectivity, and there are these complex interactions between these different neuromodulatory systems. So I think at best what we've done is we've managed to hit this system with a hammer and we've broken it. We would like to do some more nuanced interrogation and try to figure out which parts contribute to what. Okay, so this is really what I wanted to get to. This is kind of the point of my whole talk. So I think what I would love to hear people's thoughts on is if functional connectivity really is dominated by neuromodulation, what does it mean? Now, I will freely admit that we haven't proven that this has happened yet. We are in the process of trying to do some optogenetic experiments in rats to get a better idea of, if we turn activity in certain nuclei up or down, can we get a predictable response in the QPP? But let's just say, that all the evidence that points this direction really pans out. So then how does that change, how we think about functional connectivity? And I think one thing that really has come across to me is that we really should be thinking about this functional connectivity as like a snapshot of a whole brain coordinated process. It's not so much that functional connectivity between, say, left and right hippocampus, is changing because of a working memory training or something. I think we need to think about this at the systems level and how the dynamics across the system change and how these functional connectivity snapshots then reflect that overall organization. Now, my hope, what I think would be really cool, is that maybe each QPP is weighted towards some particular neuromodulatory system or some combination thereof. So maybe our primary QPP is driven by the LC, but if you have more serotonergic input, we get QPP number 2. So this is the kind of thing that we want to go and try to figure out with the optogenetics. So I think it's a really cool possibility. I have zero evidence that it's actually true yet. The other thing I think that we should consider is what else is in the bold fluctuations. So we know from task-based studies, we know that neural activity can be reflected in the bold signal. So it must be in these spontaneous fluctuations too, but how do we get to it? And maybe if we take the QPPs out and we try to look at what's left, maybe that will help us find a way forward with that. I don't know for sure that that's the answer. And I think we should try to come up with other ways to get at these small changes that we might be interested in. And then the last question that I really have is why this interhemispheric correlation is so strong still, even after we take out the QPPs. One idea that I've been tossing around is that maybe there's this sort of vascular resonance. We know that the vascular structure of the brain is fairly symmetric, and that the vasculature is very closely intertwined with neural activity and how it's affected by it and how it might affect neural activity. So maybe when you have some sort of ongoing neural activity at a particular frequency, you can kind of set up this resonance that really amplifies that and makes it show up strongly in the bold signal. But I would love to hear some other ideas as well. So just to remind you what I'm talking about, that's this diagonal line here that's between left and right areas in different hemispheres. Okay, so I'm going to stop with that. I just wanted to put up an acknowledgement slides to thank everybody in my lab, our funding sources, and all of our collaborators who have helped us so much over the years. And I hope you all have some good questions. I'm ready.
All right, Shella, thank you so much for a wonderful talk. That was great to see the survey of your work. I know it's generated a lot of interest. Actually, I had a request that we could potentially have some discussion directly, not necessarily just through the chat. And as long as things can maintain a level of civility with, the 80+ people that are remaining, I think that we could try to welcome that opportunity. So I know Amir Shmuel, you were interested in asking a question. You want to go ahead and unmute your microphone and essentially ask?
Yeah, sure. Thank you very much. Hi, Sheila. Great talk. Thank you for this very stimulating talk. And Nathan, I will try to stay civil as much as I can. So I have, several comments. And the first one, I think, is a little bit provocative, but I think that it can sharpen what is QPP relative to a network or relative to an independent component, etc. So the provocative part of the comment is that, in my view, given a network and that one area in this network fluctuates in activity up and down, then QPP can be derived from that. So it's a different description, but it reflects the fact that we have networks and that the activity goes up and down. Now, I said that I think my comment can sharpen what a QPP is. And I believe that what QPP adds to that is possibly the spatiotemporal progression within a QPP, meaning if the progression of the activity, say, goes along the gradients in a reproducible manner, more like one direction than another, then QPP describes more than just networks or independent components. My second comment is about what functional connectivity reflects. And you showed evidence that it may reflect neuromodulation from subcortical structures. And it makes a lot of sense. But I think that this is not the whole story. And the reason that I say that is that others, and my lab too, have shown functional connectivity in the resting state in a scale finer than a cortical area. In our case, we looked at areas V1 and V2 and V3 and showed functional connectivity within an area and between an area at a scale which is finer than a cortical area. And as far as I know about neuromodulation, it's not, the connections from subcortical cortex to some subcortical structures to cortex are not, do not alternate in a fine scale. They just innovate a whole area or a whole network. So I have a third comment, but I'll leave it at that, and maybe if there is time later on.
Thanks, Amir. Those are great comments. I'm glad you left off the third one because I can only remember two things at once if I'm lucky. To start with, so the first one was about what QPP can add to independent component analysis or networks. And I think you're spot on. What we see is, you would expect to see the patterns because we know that these networks are correlated. What we see from the QPP, though, is a coordinated transition between these networks and a coordinated way of moving between one network having high activation to another network having high activation, where it does go across gradients in particular ways over time in a very repeated manner. So you're absolutely right. What we add is more of the spatial component about how things change than saying that, oh, these networks are actually different than something that we're seeing before. So we think that in general, what you see as functional connectivity networks, if you do say ICA, are snapshots of which areas tended to have the same phase of the QPP. So I think that was comment one. Comment two was about the neuromodulatory input not having such fine structure. And I completely agree with you. And I think this is great. I've seen the work from your lab and from some other labs where if you look at sub areas, instead of say somatomotor cortex, you look at particular hand area versus foot area, et cetera. And you see this finer subdivision where you see more specific connectivity between areas that are alike. So I don't know as much as I would like to about the projections of the neuromodulatory system, but I agree that they're unlikely to be differentiated at that level. So one of the things we've been thinking about maybe as part of this picture is that there are seed regions that kind of drive and the activity propagates out. Whether it propagates via astrocytes and calcium waves or via some other mechanism from these sort of lateral connections, that are in the cortex. I don't know. This is something I really don't understand yet, and I'd like to try to figure out how this works. But I think there could be something else that goes along with the kind of this neuromodulatory input that accounts for the way that we see the activity move across the cortex.
Thank you.
All right, do we have any other questions at this time? All right, I'll jump in then. So Shella, I'm wondering how, I mean, this is really fascinating, like seeing these these oscillations and these patterns, and you're looking at the dominance of them. I'm wondering how much would you say those spatial topographies are driven in part by structural connectivity versus, some of these other more functional and phasic processes?
I think that's a good question. I mean, certainly the structural connectivity plays a strong role. I mean, if you look at structural connectivity versus functional connectivity, there's a lot of overlap between those matrices. But we do tend to see areas involved that are not known to be directly structurally connected with some of the other areas. So I think the answer there is pretty much the same as for any functional connectivity study, and that, it's kind of a mix. We have to have the structure. But a lot of times when we talk about the structural connectivity, again, to go back to this idea that activity might propagate along the cortex, we're talking about structural connectivity based on white matter tracks that we're able to resolve, which of course is a subset. And we know that there are connections within the gray matter itself, some of these short-term connections, some of these U connections, and other connections that are hard to resolve with DTI. So we're not getting a full picture of the structural connectivity. If we had a better picture of the structural connectivity, maybe it would, explain more of the QPPs, and maybe it wouldn't. I'm not sure.
All right, so we have a question from Vicky Morgan, who's asking, how do you think QPPs change with arousal for awake and during sleep, etc.?
So from what we've seen, they tend to become stronger and more frequent during arousal.
Okay.
Yeah, exactly. I don't have, we've gotten a bunch of small data sets on that. I don't have anything that's published that I can point to, but we've looked at it in monkeys that were awake, asleep, or anesthetized, and we've looked at it in rats under different levels of anesthesia, and so it tends to hold for those cases at least.
All right. So I'm going to ask another question. I was really fascinated by the similar topology as the gradients. And I'm wondering, something in our lab, we've started playing with gradients ourselves, and we're interested in the order in which they are present in the data. So it's often you see that default to, primary perceptual motor as the first and second gradient. What do you suppose the relationship is to the order of the gradients, which are explaining that variance, and is it the strength and the frequency of these QPPs? What are the features of the QPP that's potentially resolving those sort of gradients and their stability?
I think it's probably the strength, the fact that these are so strong. I mean, once you know what you're looking at by eye, you can almost see them in human data. It's really easy in rat data, actually, but you can almost see them in the human, at least for the really, the primary QPP. The other ones are harder to pull out. But I think, well, it's probably a combination, honestly, because it's not just that it's strong, it's that it repeats. And so that's why the averaging algorithm that we have is able to pull it out so clearly. And so I think, when we talk about strength, one of the metrics we use sometimes is just to sum up all of the correlation coefficients between the template and the time course, because that kind of takes into account both, the strength of the correlation, or how much that pattern is present, and how often it shows up that way. So I think it's a combination of those, really.
All right, so we have another question. Could QPPs be responsible for fast recruitment of brain regions required for different cognitive processes? And do you believe that disruption of these organizations could lead to poor cognitive task performance?
I do think that's a possibility. I don't have much evidence to say what happens with that yet. We've looked at the QPPs during task performance. It was in the HCP data where people did 0 back and 2 back data. But it's hard to say exactly what's happening because those blocks were very short and the QPPs are fairly long. And so we weren't getting a full QPP cycle per block. So we saw a change, but it was hard to disentangle the on and off effects from the QPP effects. Another thing though we've looked at, one of our collaborators has looked at QPPs in mice who are lightly anesthetized and suddenly given a visual stimulus. And it looks like the visual stimulus triggers QPPs. Now, whether this is because, you know, it's somehow preparing the brain for activity, or if it's just an overall change in arousal, I don't think we know. But there are some really interesting interactions with tasks that we'd like to explore.
All right. Great, thank you. So we don't have another question. I think, Amir, if you'd like to come back with your third question for Shella. No, you're still muted if you're interested in asking your third question.
Yeah, I guess I asked myself the same question while being muted. So, you know, it's not a question. I just agree that the interhemispheric correlation is a puzzle and the mechanism is unclear.
Do you have any thoughts on what it might be?
Not really. I've been thinking about that for 5 years. So I don't know.
All right. So, Shella, I was wondering if you, the tools that you've developed in order to identify QPPs, is this sort of, are these tools available to people to start exploring their own data and these features? And can you talk about that at all, as well as potentially, you know, in the realm of fMRI, like what's good data to look at? Do you need like high temporal sampling and whatnot?
Sure, yeah, absolutely. The tools are available. They have now been built into the CPAT pipeline, if y'all are familiar with that. So I always forget what CPAT stands for, but it's something about, you know, pipeline for analysis of connectomes. Mike Millam, Cameron Craddock, or some of the lead people on that. So it's built in now, so you can do your analysis if you use that pipeline. We also have some nice MATLAB code that we're happy to share. So if you want to run it through in MATLAB, we can pass that on to you. In terms of what kind of data you need, you know, I think anytime you're looking at dynamics, it's nice to have a short TR and a fairly long scan. But that said, it's not always necessary. It probably depends on what you want to see. If you wanted to see differences between groups that might be fairly subtle, it's probably more important to have, you know, shorter TR or longer data or both. But if you just want to be able to see a QPP and maybe regress it out to look at functional connectivity, we've gotten quite decent data at the individual level for Human Connectome Project. I think if you go to something with a longer TR, like 2 or 3 seconds, we've managed to pull them out, but then we need more scans. So there's kind of this trade-off between how fast you image and how many total images you need to get a good estimation of the pattern.
All right, great. Well, Shella, thank you so much for being here today. We really appreciate you sharing your work and your science with us. And thank you everyone for showing up today. It was great to have so many in attendance around Montreal and the rest of the world. So please tune in next week if you're interested. We're going to have Dr. Jessica Payne from Notre Dame joining us, talking about sleep and memory and its relationship to the functional organization of the brain. Although we have one more question that slipped in, and there's a question of whether or not you see any changes in QPPs signaling with normal aging. Shella, if you could answer that before we sign off.
Well, we haven't looked at it specifically yet. We looked a little bit at Alzheimer's, and there are some hints of changes, but that was something done by a rotation student, and we haven't been able to go through and really thoroughly explore it yet. So I think it should be there, honestly.
All right, great. Shella, thank you so much again, and we look forward to hearing about your work again in the future.
Well, thank you so much. It was great to talk with you all today. Have a good one.
You too. Bye.